So, yesterday we talked about ECM, the protein part, that where I have discussed first this elastin, laminin, fibronectin, and then extensively on collagen. At least in collagen you must remember the triple helical structure, quarter staggered arrangement, abundant protein of the extracellular matrix of the and also of the body, and diseases associated with defective formation. Today rest part I will be talking. Yesterday I told you that between the cell there is a gap, the extracellular part. That extracellular part is maintained by the extracellular matrix and they perform unique functions. Yesterday I discussed. Again today I will tell a little bit about it. This extracellular matrix is required for the normal growth, differentiation, development, maintenance of tissue structure and optimal cell functioning, and also keeping the cell in position. Because when there is a tumor or cancer, usually the cells also spreads, and the spreading occurs because of the damage of the extracellular matrix. Otherwise, cell cannot move. So therefore, all the structures which are anchoring the cell to the intracellular matrix or between the cells, that must be loosened somehow. Otherwise, that even if the cancer cells are there, they will not be able to move. So as you know, the cancer cell spreads, and the spreading is possible only because of those protein structure and the structures of the extracellular matrix if these are damaged. And cancer cells do have the unique property to secrete enzymes which basically destroy the components of extracellular matrix. Therefore, they get detached and then they spread, they enter into the lymphatics, they enter into the bloodstream also. And also as we age, there is a visible change in our body, particularly during aging that I will discuss also at the end, what are the changes that occurs, because cells are tightly packed to each other by the proteins. Now as we age, you know the elasticity of the skin also becomes lax, and the skin strength is also weakened. And this is because the proteins which are present between the cells and the extracellular matrix gets damaged, and that's a normal aging process. Now, if you know the biochemistry part properly, then you must be by this time aware that to maintain the beauty of any person, many anti-aging agents are used. The basic attempt of all those agents are to inhibit the destruction of the extracellular matrix somehow, so that the appearance of the body or the skin remains intact and you do not look aged. Same thing is true for the during wound healing also, and you will see during wound healing, as I will discuss also at one slide, that what are the changes that take place, because that is almost a rapid action type of extracellular activity. The body is damaged, the wound is there, it needs to be restored as fast as possible. So all the functions which normally are present or operating in the normal body system gets too much accelerated. And in the process, perhaps you have also experienced that during your wound healing, there is not absolute formation of the body structure akin to what it was there with the original structure. There might be some increase, and in course of time, there is remodeling of that entire part to restore as near as normal. but this may not be possible always because of the defect, because of the extensive nature of the damage what we have undergone. So, I told you yesterday also that the various components of this extracellular matrix basically keeps the cell in position, and they allow the cell to function by maintaining its not only its structure but accommodating the interior of the cell with outside of the cell, and there is a continuous, this structure is continuous that I have explained yesterday. So, whatever you are seeing between the cells, you see there are all the areas are not present, all the areas are not covered with fully cells. So, there are spaces. So, these spaces are basically intercellular space or extracellular space, and where these extracellular matrix components are present. Bone, yesterday I could not cover. Bone is a dynamic structure. Although we are maintaining our body physical structure almost same thing is true with bone, but there is a continuous wear and tear, and old cells or senescent cells are getting destroyed, and they are getting also replaced. So, bone is formed by two elements, one is organic and another is inorganic part. That by this time I think with the help during the class of parathyroid hormone, you have learnt a lot. But the major component of bone is collagen type 1, and it is synthesized by the osteoblasts. So, osteoblasts synthesize collagen type, and it constitutes 90% of bone protein. Type 4, 5 is also there, and there are other non-collagen proteins like say plasma proteins of different types: osteopontin, osteocalcin, osteonectin, bone shallow-protein, bone morphogenetic protein, osteoproteoglycan, because all these proteins have unique functions to carry out so that the bone structure is maintained, bone is mineralized properly and remains in its position. And among the inorganic part, the calcium, 90% of the body's calcium is stored within the bone. Other components which are present, there are sodium, magnesium, and phosphorus. And in the bone, calcium is not remaining just like calcium; it remains as hydroxyapatite insoluble crystals. So that is the reason why calcium cannot be removed so easily from the bone, and it acts as a basically reservoir of the body's calcium. So when there is a requirement of calcium in the body, say PTH hormone is released that activates the osteoclasts and bone resorption occurs, and that is the reason why we have an increase of the calcium. And when excessive calcium is also formed, then calcitonin hormone is released, which decreases the bone formation, and it helps in bone deposition. So major cells in the in the bone are osteoblasts, osteoclasts, and osteocytes. Osteoblasts are primarily responsible for formation of osteoid. You see, this is the osteoid, and here the type 1 collagen is synthesized, and it forms the matrix of the bone, where the trapped cells and mineralization takes place here only. Now, osteoclasts are this cell, they help with the bone resorption and releases calcium. So, normally alkaline phosphate is there, it helps in releasing phosphate, and it helps in mineralization. But by lowering the local pH, the hydroxyapatite crystal is solubilized, and that is the reason why calcium gets released. Now, osteoblasts, when they are mature, they become osteocyte, and they are the differentiated form of osteoblast, and they remain within the bone matrix and maintain its structure. So, bone mineralization usually takes place by osteoblasts, they control mineralization. As I told, that synthesize type 1 collagen and also type 4. and it is regulating also the passage of calcium and phosphate across the cell because of the as per the requirement. Second, alkaline phosphatase, bone alkaline. You know that there are 5 alkaline phosphatases: one liver, intestine, placenta, bone, and IPKBL. So bone alkaline phosphate is there, and this function is to generate phosphate. So mineralization is first evident in the gaps between the collagen fibers. Collagen is also synthesized. So there are gaps in the collagen. So basically, the calcium hydroxyapatite gets deposited in between the spaces of the collagen fibers, so that they are tightly packed. So bone shallow protein and osteopontin, as you say, these are the non-collagen proteins. They act as a site of nucleation, because somewhere this has to be the site of nucleation must be there, around which the bone mineralization will take place. So it forms a scaffold basically, just like other structure. Vitamin D helps in calcium absorption from the gut, and PT, as you know, that it regulates the calcium level both by, and it is having an action of both on osteoblast and osteoclast depending on the calcium level. So when there is osteoblast, that means there is basically a bone formation, and when there is an osteoclast, then that means bone resorption. Calcitonin inhibits the action of osteoclast. Estrogen is very much important in female. You see the role of estrogen. Estrogen basically on the osteocytes, they inhibit bone remodeling, and osteoclast decreased bone resorption, and osteoblast maintenance of bone formation. Now this, because of this estrogen presence during the reproductive part of a woman's life, usually osteoporosis doesn't occur. Rather, osteoporosis doesn't occur in an otherwise normal individual having a normal reproductive function because of the estrogen level. Now, after menopause, because of the estrogen effects are not available, the bone gradually starts undergo bone resorption and osteoporosis. So osteoporosis is a little bit more common in women than in men. So after menopause, it is advisable for all the women to take some amount of calcium and vitamin D to ensure good amount of calcium, so that the osteoporosis is minimized as much as possible. So cartilage, type 1 collagen is present in bone, type 2 collagen is present in cartilage, that you must remember. Type 2 is cartilage. So yesterday I told there are 3 types of cartilage. Already you know in the, I think it has been taught in the anatomy class, the hyaline, elastic and fibroelastic type. Of course, the fibroelastic type contains type 1, and there are different types of proteoglycans that I will tell in a short while. One of them is an aggregan. This is a very complex structure, and chondronectin. These chondronectin and aggregan are the this is a proteoglycan, and the chondronectin is a cartilage matrix protein, and they are present in the cartilage. Glycoprotein and proteoglycans. These, during your carbohydrate class, I think this has been discussed during as part of GAG, that is glycosaminoglycans or mucopolysaccharides. You know the name of hyaluronic acid, heparan sulfate, keratan sulfate, darmatan sulfate, heparan sulfate, chondroitin sulfate. These are the basically glycosaminoglycans. They are required for extracellular matrix formation. A glycoprotein, and the difference between the glycoprotein and proteoglycan is by and large with a percentage that is in glycoprotein. Basically, mainly it is a protein part, and to which an oligosaccharide chain is attached. So carbohydrate content is less, usually less than 10%, but in proteoglycan the carbohydrate content is more than 10%. It can be as high as 95% as well. So predominant part of proteoglycan is basically a carbohydrate, while glycoprotein part it is mainly protein. So, what is this thing? You try to understand this structure. You see, this is a core protein. How this proteoglycans is formed? This is a core protein, and this is a linker protein. Around this core protein, these glycosaminoglycans are attached to a covalent linkage, and this is also a glycosaminoglycans, that is hyaluronic acid. This core protein is attached to this GAG through a linker protein, and they make a structure which is something like a brush-like, and because of this a large structure, they occupy a good amount of space within the extracellular matrix, and because of this structure, they also draw water and maintain osmotic pressure, and also maintains, therefore it maintains the turgor of the tissues. As you see, if you can see the skin and the skin spot or any tissue, you can see the turgor nature of that particular tissue, and it differs from tissue to tissue. So, proteins that contain covalently linked glycosaminoglycans, and it's a repeating units of disaccharides that you know it is composed of one amino sugar and one uronic acid. Either it is a glucosamine or galactosamine, and uronic acid may be in the form of glucuronic acid or ioduric acid. Now, there are at least 30 different types, of which a few are important, that is syndecan, beta-glycan, biglycan, agrican. These you have seen, perlecan, fibromodulin, and this structure what I have discussed has been depicted here in the in a picture form. Now, if this structure is seen in an electron microscopy, you see the typical structure looks like this, as if it's an image of what I have said, and this is called a bottle brush like of appearance. So, glycosaminoglycan, this proteoglycan appears as a bottle brush like appearance during electron microscopy, and you can see because of this structure, as I was telling, that this is a huge structure, and it draws water, and it maintains a good amount, occupy a good amount of space within the extracellular tissue, and it maintains its turgor of the tissue also. The difference you note down, if you want to note down, that what is the difference between proteoglycans and glycoprotein. See, proteoglycans is a multi-domain core protein, and in which this GAG, what I have said, is GAG 1 or it is also a GAG. Hyaluronic acid is also GAG. There might be a separate other GAG which might be present. It's a multi-domain structure. Glycoprotein, as I told earlier, this is a oligosaccharide chain which is covalently linked to protein. And location is mainly proteoglycans are in the connective tissue, that is in the extracellular matrix or the ground substance. What do you say? The other name of extracellular matrix is ground substance or connective tissue. In contrast, glycoproteins are present usually on the cell surface, and they act as a receptor, and they help in cell-to-cell recognition and signaling. But glycoproteins may or may not be negatively charged. Examples of the proteoglycans are basically all the GAGs, that 7 types of GAGs you must remember: hyaluronic acid, heparin sulfate, chondroitin sulfate, dermatan sulfate, and keratin sulfate. There are 2 types, type 1 and type 2. Glycoproteins, example, leucine, like transferrin, which I think there is a question on iron absorption, and immunoglobulins. These are the examples of glycoproteins. So, proteoglycans are major component of extracellular matrix, and I told you it is covalently linked to a specific protein core, and there are 3 different types. One type is small leucine-rich proteoglycans. There are 18 types. So, functions of proteoglycan, you note down all these points, because this may not be available in your class, in your book. I have somehow combined so that you remember easily. Number one, it determination of structural organization of the ECM in the tissues. How? It is present in every tissue. It is not that it is present in a specific organ specific. So, I told you that it is filling of the space because of the large structure. So, they are associated with each other. Suppose this is one GAG, this is another GAG. So, they might be associated with each other here. They might be associated with other protein molecules. So, specific interaction is there for all the proteins present in the intracellular matrix, so that the bone structure is altogether different, liver structure is different, retinal structure is different, cornea structure is different, and because the GAGs are specifically attached to proteins, so that determines the structural organization of that particular tissue. Second, contribution of characteristic target of various tissues. You, by this time in the anatomy, you have seen or in the living body, you can see the texture of the liver or the target of the liver, or meat you have seen in the kidney, liver, heart. If you see the tissue, it is not same. So that is called characteristic target of the tissue, and this is present because of the polyanions present in the GAG. They are negatively charged, so they draw water, and they can exert osmotic pressure, and because of the osmotic pressure, just like the albumin or plasma proteins, they draw water in the extracellular space. So, extracellular matrix is a gel-like substance. It is not a hard substance, as I explained yesterday. Now, again, because of the large structure, it acts as a sieve, means it does not allow the larger molecules to pass through, but allows smaller molecules to pass through as diffusion, or they can pass throughly. So, they act as sieve because they can gel at low concentration, and again the occupancy of large volume of matrix because they can absorb a huge amount of water, and their huge macromolecular aggregates are there, which are not present in other substances. So, glycosaminoglycans, just a repeat thing, nothing more than that, all of you know. So, only thing is the homopolysaccharides, you know that starch, glycogen, starch we take and glycogen is present in our body. Now, glycosaminoglycans, the presence of charge group that attracts molecules and produces a viscous solution. That is the reason why extracellular matrix is gel-like a viscous solution. Mucopolysaccharides, they are also named as GAG. They, when they combine with protein, they are called mucoproteins. So, just to remember, to help you to remember, there are two things. One is N-acetyl-glucosamine and N-acetyl-galactosamine. Remember only these two things, chondroitin sulfate and dermatol sulfate. The component is N-acetyl-glucosamine, and the other component is there in the hyaluronic acid, heparan sulfate, heparan, all with 3 H on the first. They are basically with N-acetyl-glucosamine, not galactosamine. That you must remember, and to which keratin sulfate is there. The difference between the keratin sulfate does not contain any uronic acid. This is a short MCQ question. Hyaluronic acid and heparan sulfate, if you look at the composition, they are the same. Only difference is that hyaluronic acid does not contain any sulfate group. That is the unique properties of hyaluronic acid. And keratin sulfate, there is no uronic acid, although we say that a GAG is composed of an amino sugar and an uronic acid. But keratin sulfate, although is included in the GAG, but they do not contain uronic acid. So, what are the functions of hyaluronic acid? As I told you, these GAGs are basically repeating disaccharide units. So, they are present in the hyaluronic acid is present in the synovial fluid and vitreous humor, and they perform lubricant because joint space, the has to be lubricated, otherwise the joint will not move. So they will act as a lubricant, they act as a shock absorber, something akin to the CSF, you can say, that there is some amount of fluid is there, so it acts as a shock absorber. It helps also in cell migration and wound repair, that you will see, because the short while during changes of ECM in wound or during the wound healing, basically. Wound repair, they play an important role. Cell migration is important because in cancer, the cell migrates because the hyaluronic acid, there is rapid spread of hyaluronic acid in the surrounding tissue, so that they get detached. So exceptional property, there are two exceptional properties of hyaluronic acid you must remember. One is it exists in the ECM without covalent bond. Normally all the GAGs are bound to protein by covalent bonds, but hyaluronic acid is not linked. This hyaluronic acid here is not linked to covalent, but it is a non-covalent bonding, and it does not contain any sulfate group, either as O ester or as N sulfate. Heparin, all of you know, it's an anticoagulant. Chondroitin sulfate is another important structure that is present in the connective tissue, cartilage, bone, cornea and skin. So they maintain the shape and structure. You see, the keratin sulfate doesn't contain uric acid, and it's a repeating units of galactose and beta 1,4-N glucosamine. It is present in the cornea and tendons. Now you see, if there is a sulfation, so if there is a defective sulfate, normally if you look at the cornea, it is very much transparent. Now if there is a defect in the sulfation, then you see the dotted spots, the white spot. So that is a macular corneal dystrophy. So that will result if there is a defect in sulfation. Similarly, as I told you, it maintains the shape of the structure. So normally cornea is convex. Now here the cornea, look at the cornea, it is somehow a conical. So it is called keratoconus. So defective formation of keratin sulfate, if you just look at the eye by putting a torch only, you will be able to diagnose that this is a case of keratoconus. Of course, the other causes of keratoconus are there, but one thing you have to remember, that it can be due to defective formation of keratin sulfate. So the person will be having visual problems, simple. So darbeton sulfate, as the name indicates, it is present in the skin, maximum amount is present in the skin, also present in the blood vessels, heart valves and tendons. Again, it maintains the shape and structure of the tissue. Now you note down all these points, changes during aging, because in your syllabus, the point on ECM is basically role of ECM in health and disease. So this might come as a question also. All these 3 points which I will be discussing. One is changes of ECM during aging. Why I have elaborated the class so long? Because without having a formal introduction about what ECM is all about, you will not be able to understand this part. You see, aged tissue are less elastic, less rigid and weak. And why this will happen, as I told you in the beginning, that normally the cells are attached to each other tightly and also with the extracellular matrix. Now that part has to be damaged somehow. So first, there is a decrease in the junction protein. So if there is a decrease in the junction protein, that means cell gets loosened. Second, if there is a gaps, then at one point of time, if the process goes on, then cells will finally, will not be able to attach to each other. So there will be gaps at the junction between the cells, and enhancement of the matrix metalloproteinase mediated degradation. Now, this enzyme is very much important because during wound healing, this particular enzyme is activated, so that there is a matrix formation. In aging, there is enhancement of the matrix metalloproteinase mediated degradation. Not this function of this thing, it is mediated degradation is activated, and this is the resulting in thinning of basement membrane. In contrast, during wound healing, we require basement membrane formation. So, therefore, that matrix metalloproteinase enzyme needs to be activated. And senescent fibroblasts, as you age, there are many biochemical changes, and at one point of time we die. And why we die, there is a big book, there is a book written by Venki, who was basically awarded Nobel Prize, Fibronectin, why we die. And the entire details of aging process have been written in that book. So, senescent fibroblasts express more cytokines, MMPs, and plasminogen activator, and they also produce reactive oxygen species of the mitochondria. When there is a reactive oxygen species, that means this will damage the structural component of the body tissue. So, chronic inflammation is there, so destruction of integrity of elastin network and basement membrane. Now, yesterday I discussed there is a cross-linking of collagen after formation. Now, that inappropriate collagen cross-linking because of the reacting oxygen species, and as we age, there is exposure of continuous UV rays and enhanced glycation that causes stiffness of the tissue. Changes of ECM during wound healing. Another problem, this is what is the role of ECM in disease. So, fibrogenic mechanism is activated because you see, as I told yesterday, that collagen is formed by the fibroblast. So, therefore, fibrogenic mechanism has to be very much strong, very much activated, otherwise collagen formation will not be there. So, there will be activation of fibrogenic mechanism, and the monocytes which are present within the circulation enter into the tissue spaces when there is an inflammation, when there is a damage. So, they turn into macrophage, and then macrophage basically releases growth factors. Now, we come, I told you matrix metalloproteinase enzyme which forms the basement membrane. So, matrix growth factors is there so that the cells will grow. So, matrix metalloproteinase and cytokines, though they favor angiogenesis because you see that structure contains blood vessels. So, the restoration of the circulation of that entire area will be required. So, angiogenesis will be to be there. Then fibroblast migration, proliferation, and then its activity also. So, trans differentiation of fibroblast into myofibroblast, because if it is a muscle tissue, then the muscle tissue has to be formed. So, ECM deposition, collagen bundle synthesis, as I told you, because of the fibrogenic mechanism, collagen bundle synthesis will result. As strengthening of the tissue and remodeling, as I told you, during wound healing, the wound initial stages is not the same as of a normal structure. It might be usually raised or increased. So, after remodeling, the restoration of normal tissue is maintained as nearly as possible. Now, restoration of tissue homeostasis and resolution of fibrosis. A role of proteoglycans in major diseases, cancer. As I told you, normally the cells do not move from the tissue. Now, in cancer, there is movement of cell from one place to another. That is the reason why cancer spreads, and there is a lack of adhesiveness of the cells, between the cells you can say, or among the cells, which is because of decrease in the heparan sulfate. And the spread is because, as I told you in the beginning, there is great increase in the hyaluronic acid. So, that dissolves that extracellular matrix and causes the tumor cell to migrate, atherosclerosis. Darmatan sulfate is present in the intima of the arterial wall and that causes deposition of plasma LDL, that is basically binds with the plasma LDL. So, darmatan sulfate in the arterial smooth muscle present, they also proliferate and they make an atherosclerotic plaque. And the mechanism of formation of atherosclerosis by LDL cholesterol is not only by LDL cholesterol per se, but through darmatan sulfate. Arthritis. Normally, our body tissue we do not mount any immune response, but at times because of the error in immunity, our body tissue fails to recognize our body cells as our own and they mount an immune response that is called autoimmune disease. And here you can see glycans which are normally present in the body as a part of normal component, they act as an autoantigen and they mount an immune response in arthritis. And in cartilage, there is decrease in the chondroitin sulfate. It's a component of cartilage and there is increase in keratin sulfate or hyaluronic acid. So, basically, normally these glycosaminoglycans are formed. These are disaccharide, repeating disaccharide units. So, there must be a mechanism for formation. Now, once these are formed, they are also required to be degraded. But if there is a defect in degradation of the glycosaminoglycans, then there will be storage, just like glycogen storage disease, because that glycogen, whatever you have formed there, glycogen is not being broken down because of the lack of enzymes. So, here again the mucosaccharides you can see these, what you have to remember that these diseases are the name of the enzymes which are absent. Like here you see the hurler, if you cannot remember all these things, at least remember three things. One is hurler, one is hunter and one is slide. See hurler, the deficiency is L-iodide and hunter it is iodinate sulfate and slide disease it is beta glucuronidase and now it is available in the market, it is an FDA approved drug. Now treatment is there, it says all these things are rare diseases. Now if you look at the hurler disease and this type 1 and type 5 scaly, both are having L-iodinate deficiency. But if you look at the clinical picture, there is a mental retardation, but here there is no mental retardation. Here there is a skeletal deformity, but there is mild skeletal deformity. Here both darmatan sulfate and heparan sulfate is present in the urine, but here only darmatan sulfate is present in the urine. So you make a chart and you try to remember all the diseases. There are Sanfilippo, there are 4 types A, B, C and D. Mercuos, there are 2 types, one is A and B. Scaly's disease is again iodinate deficiency. So, because of the inability of to degrade those GAGs, these GAGs are present, gets accumulated within the body. That is the reason that these diseases are produced. So, you remember that this first 3, what are the MPS 1, 2 and 7. You cannot remember all those things. Just look at the picture, the Hurler syndrome, because those who will be opting for pediatrics or the gynae during delivery after birth, in your lifetime, you might get a case of this, all these rare diseases, because these are not so common. So, at least I have not seen, and I have inquired from others also, they have not also seen, but there is a chance that probability that since it is described, so you may get a chance to detect this disease, provided you know it. So, from the appearance you can diagnose this case. So, this is Hunter syndrome, the Hurler syndrome, this is Sanfilippo. They are looking apparently normal, but they are basically defective. mark you see the look, all these things appears to be the same. Now you see this is a wheelchair bound. Look at the fingers, you can diagnose. Now it may happen at times that there will be a monozygotic twin or dizygotic twin. Whether it is a monozygotic twin or dizygotic twin having the same disease, you will be able to differentiate between the two. Actually, I got one case in the OPD where there are twins. Now, we wanted to know whether it is a monozygotic twin or dizygotic twin. From the physical appearance, it was a little bit, facial appearance is not same. I have the picture. Now, we looked at the toe. All the toes, the fifth toes are not fifth toes, there are 6 toes are there, and fifth and sixth are joined together, and that indicates basically that is a monozygotic twin. So, this is Kelly's disease, MPS 4, MPS 6 is there. You see horrible things, I mean, to have a child in a family. The family parents are so unfortunate. What are actually SLY syndrome. Fortunately, beta glucuronidase enzyme is available now. This can be treated, and William S. Sly, who discovered this thing in described this disease in 1973, is still alive, and the treatment was done at the Department of Biochemistry and Molecular Biology under his care, and that's why he is a famous person. Slides disease has been named after him. So, beta glucuronidase deficiency in Sly disease may come as a MCQ, particularly Hunter syndrome, and the Sly disease. At least you try to remember these 3 things. So, summary, as I told you, that is I am closing. So, it is a repeating disaccharide unit. Remember. Every I list 10 times I have told this thing. So, containing uric acid and amino sugar, and I mean exceptional property of hyaluronic acid, as I told, it does not contain sulfate, and creatine sulfate does not contain uric acid, you must remember. Major glycosaminoglycans are 7 types that you must be able to tell during your examination at a stretch. The 3H you remember, heparin sulfate, and hyaluronic acid, keratin sulfate 1, keratin sulfate 2, dermatan sulfate, choronoidin sulfate, bas, finish. At a stretch you must be able to tell. So, they are synthesized, as I told you, they are synthesized by sequential action of glycosyl transferase, epimerase and sulfotransferase. So, they are synthesized, but they are broken down by the lysosomal hydrolysis, because these needs to be clipped, they are attached to each other through a glycosidic bond. So, that bond needs to be hydrolyzed. So, if there is a defect in the hydrolysis, then only this mucopolysaccharide will accumulate, and it will result in the formation of mucopolysaccharides of different types. There are 7 different types. You must make a chart, and you have to remember at least lifelong, I would say, at least till your examinations are over. The questions are usually, I mean, at least 1 or 2 questions will be there from these diseases. So, they are bound to various protein as linker protein, as I told you, and attached to the core protein also, and they form many functions, and genetic deficiency of the enzymes which degrade causes disease called mucopolysaccharidosis. So, that is the end of the class. If you have any question, you can ask any question, because there is no more discussion on this topic next time, and you have to read. At least try to finish today itself, otherwise you will not be able to get time, and you will forget what I have all told. I have been repeatedly detailing. Even before examination, if people do not come prepared, you do not read properly, and that must not happen, because now your third internal assessment is just another 2 months time, and final examination is knocking at the door. So, do not waste your time. Again, I am telling you, take these subjects very seriously, because I don't want to see anyone to fail. Whatever performance you have made in a internal assessment, forget about that thing. All those failures or less marks will not matter at all during your final examination. So, again, as I told in every class, that you read thoroughly and try to discuss among yourself, otherwise you will not be able to remember. It's not that easy. You have to repeatedly tell. Even at our stage, we cannot remember all those things. We need to read. All of us need to read. Don't think that all the faculties, whatever are present, they are simply sitting idle. They all read books. They cannot remember. It is not possible, humanly possible to remember each and everything. Even I am telling you, see, whatever you have passed in a Madhyamik or has a secondary level HS, I think hardly if the same question is given to you, you will score nearly 5 to 10% of the marks what you have got, because that means that much knowledge is with you. The same thing is true with me. If you give the same biochemistry question paper to me, I will be able to get the marks which I got during your past professional examination. That means that much information I could not still remember, and that happens. That's why continuous reading is required, and you discuss among yourselves if you cannot remember alone, and practice writing also. That is what is required. Okay? Any question? Any question? I'd love to take some questions and want to answer. Again, I am telling you, I will be very much happy if I cannot answer, and that will be a food for thought for my own reading. Okay? All the best.